Hello everyone! Today we're going to talk about alternate splicing in relation to sex determination in Drosophila. First, a quick reminder of the central dogma of biology. DNA encodes messenger RNA, which in turn codes for proteins. However, there are some key differences between these processes in prokaryotic cells, which carry out transcription and translation in parallel, versus eukaryotic cells, which carry out transcription in the nucleus and translation in the cytoplasm. In eukaryotes, mRNA processing and export must take place before an mRNA transcript can leave the nucleus and undergo translation. As part of this RNA processing, mRNA needs to be capped on the 5' end, non-coding regions, aka the introns, must be spliced out, and the 3' end of the transcript needs to be cleaved and polyadenylated. This mature mRNA can then be exported out of the nucleus and into the cytoplasm to next be translated into proteins. However, some mRNAs are able to be spliced in multiple ways and can encode different protein products. This is known as alternative splicing. This phenomenon is an important part of sex determination in Drosophila. In Drosophila, sex determination is all about the ratio of autosomes to sex chromosomes. Flies have 1 or 2 X chromosomes and 2 sets of autosomes. Male flies have 1 X chromosome and 2 sets of autosomes, a ratio of 0.5. Female flies have 2 X chromosomes and 2 sets of autosomes, a ratio of 1. The products of 3 genes in the sex determination pathway of the fruit fly are subject to alternative splicing. Sex-lethal, a splicing repressor, transformer, a splicing activator, and double-sex, a transcription repressor. Let's start with the default pathway, male pathway. So when there's a ratio of 0.5, this pathway is triggered. For the sex-lethal gene, splicing factor SPF45 binds to the 3' splice site and promotes inclusion of the 3rd exon, which contains a premature stop codon. So, sex-lethal is transcribed but results in a non-functional product. For the transformer gene, splicing factor U2AF binds and promotes splicing of the extended version of exon 2, which also includes a premature stop codon and also results in a non-functional product. Therefore, both of these protein products, sex-lethal and transformer, are non-functional in males. In the absence of these factors, in the double-sex gene, the 3' splice site upstream of the 4th exon is not recognized, and therefore exon 4 is skipped. This results in the male version of the double-sex protein, which promotes male development in flies by repressing the expression of female differentiation genes. The female pathway is a bit more complicated. In females, where the ratio of X to autosomes is 1, the sex-lethal gene transcripts are alternatively spliced so they produce a functional sex-lethal protein, which is a splicing repressor. Sex-lethal autoregulates its own splicing by binding and interacting with SPF45 protein, inactivating it, and leading to exclusion of the 3rd exon and creation of a functional sex-lethal protein. Sex-lethal is also involved in the alternative splicing of the transformer transcript. Sex-lethal binds to the splice site U2AF would typically bind to, blocking U2AF from accessing the site, so it instead binds to an alternative distal splice site in exon 2, resulting in the inclusion of the shorter version of the 2nd exon without a premature stop codon, and thus creating a female version of the transcript that encodes a functional transformer protein. When it comes to the splicing of the female double-sex transcript, the transformer protein recruits 2 exonic splicing enhancers, TRA2 and RBP1, and leads to the inclusion of exon 4. Alternative transcript cleavage and polyadenylation remove the last 2 exons, and the resulting transcript encodes a female version of the double-sex protein, which promotes female development by repressing the expression of male identity genes. And so, it is the ratio of autosomes to X chromosomes in Drosophila that drives the alternative splicing patterns of 3 key sex determination genes and results in male and female fruit flies. Thanks for listening!